The Laboratory for Optical and Computational Instrumentation at the University of Wisconsin-Madison is founded by the National Institutes of Health in conjunction with other funding sources. It has a deceptively simple mandate: develop tools and techniques to peer deep into living tissues and image the complex world inside a living organism. The staff at Loci doesn't build microscopes and then go looking for researchers to use them. They look for novel research projects and build microscopes to obtain the necessary images. Each microscope is a work in progress, constantly being improved to better suit the needs of researchers working at the facility.
What's neat about our group, I think, is that all our biologists, even though they work on very different models, you have some that work on, for example, cell fusion and stem cells, some that work on C. elegans and division, some that work on why do stem cells differentiate to why breast cancer progresses. They all have one thing in common. They all want to look at a dynamic process that's cell-based in real time and watch that in the least invasive way possible. So you want to look at things 3D, real time, and watch what's going on, all without the cell knowing we're there. And that's our dream.
Historically, a major goal in microscopy was the ability to distinguish certain types of cells from each other, or even to distinguish certain parts of the cell from other parts. The cutting-edge loci microscopes have been engineered to provide a wide variety of techniques for making these distinctions. On each microscope, beams from special long wavelength lasers interact with dye molecules in the cell called fluorophores that absorb the incoming laser and glow a different wavelength in response. Fluorophores are normally introduced into the sample from the outside or grown inside the tissues directly using genetic manipulation. In certain cases, however, cellular proteins are natural fluorophores that glow under the laser. This autofluorescence was long thought of as a nuisance because it can add background noise to the resulting image. But the loci group found that it can also be used to differentiate, for example, normal cells from cancer cells.
That's right. So in our body, things like collagen, tryptophan, elastin, NADH, these are all naturally fluorescing things. Quite exciting from a clinical point of view, but from an imaging point of view, very hard to distinguish. It's background noise. And so what we wanted to do was something I call akin to a fingerprint. It would be like trying to distinguish myself against 4 other people, and with imaging, traditionally, you only had the ability to tell us apart by the color of our shirt. And so if we're all wearing blue shirts, you'd be out of luck. But normally in a lineup, you have the fingerprint, you have the height and the weight of the person, and we want to do the same thing in imaging.
Here are other identifying factors, or dimensions, for microscopy: space and time, spectral wavelength, fluorescence lifetime, polarization. By combining these techniques, the loci investigators hope to find a signature for each different tissue.
In the biological sciences, it's often the case that smaller discoveries can be pieced together in unexpected ways for new discovery, and that's really the case in our lab. We knew that NADH glowed, for example. We knew that collagen glowed in our bodies. But we found that with using a method, this imaging method of multiphoton microscopy that could collect spectra, lifetime, polarization, harmonic, all these properties you can't see with your eye, we could really tease out new ways to understand breast cancer.
Once researchers began applying the tools available at loci to image breast tissue, they noticed the fluorophore fingerprint of the NADH enzyme changing as tissues became more crowded with cells. This was significant as it allowed for a non-invasive way to track metabolism and potentially the state of the cancer.
Well, that's the exciting thing about, I think, our group is a lot of people often misunderstand loci. They think that we're a group of experts that then collaborates with biologists. But instead, you can think of us more as an infrastructure where we bring in collaborations that include people that have problems from computer science, from engineering, and others with the biologists trying to figure out how to do this.
Improving data analysis will be an important focus for the future of the lab. Expanding collaborations in the fields of computational and quantitative analysis will enable loci to extract more of the concrete results that have made their microscope images much more than just pretty pictures.